Okay.
All right, beautiful. So go ahead with the questions. I'm going to answer real quick, and then I'm gonna ask James to do a little PowerPoint presentation, show the cells and some equipment and et cetera, et cetera. So
That would be great, actually. Thank you guys very much again. So, first of all, we wanted to start with very basic questions, like which are the main diseases or conditions that can be treated with stem cells therapy?
well, the most research for diseases treated with stem cell therapy therapies are neurodegenerative diseases. ALS, multiple sclerosis, Parkinson's, Alzheimer's, even as far as spinal cord injuries and diabetes and some others. But those are the main. Currently, right now, between University of Phoenix, James, his professor, and us, and even the UCLA, which I don't understand why they're not here. We're doing heart conditions and brain conditions. We were able to actually differentiate stem cells into cardiocytes and neurocytes. That means cardiac cells and brain cells.
Okay, and are these conditions or diseases can they be treated else like with another procedure, or is there like a specific disease or condition that can only be treated with stem cells right now?
And nowadays, with big pharma and all the medications, to answer to your question, no. I mean, there's only medications for it. Drugs, and this is cellular therapy. That's a completely different world. With through cells, we can regenerate tissue, as of compared to medications. Big pharma, they only prescribe drugs to try to make the patient's lifestyle better, but they don't really regenerate it.
Yeah, so you would say that what Big Pharma is doing right now is a temporary solution, rather that stem cells therapy and treatment is a solution to the problem itself.
Correct.
All right, thank you. So then the next question would be that where can we find stem cells in our body and how are they identified?
In our body, human body has got stem cells pretty much everywhere. The most way we can find is bone marrow and fat. Although if you extract 100 million stem cells, for instance, from bone marrow, there's only 1 to 3% top of mesenchymal stem cells, the ones that we're looking after to either differentiate to other cells or use them for specific diseases and so forth. Nowadays, we culture expanding mesenchymal stem cells from umbilical cord tissue. Healthy donors. There's a layer on the umbilical cord called the Wharton jelly, and even the cord tissue itself. We can culture expanded cells and use pure mesenchymal or some other types of cells that we can differentiate from.
All right, you kind of got ahead of me on that one because the next question was going to be how do you guys isolate and then harvest the stem cells?
Well, quite frankly, we're not using bone marrow or fat or any other, even blood. Sometimes we use blood, but there's limited amount of cells on those tissues. Although umbilical cords are a really good source for stem cells. So we isolate the microbiologists, molecular biologists. Through in a lab, we can isolate the tissue and put them in a culture media in a specific CO2 environment with antibiotics and culture media itself, and make billions and billions of cells.
Okay, got it. Thank you. And then can people donate like eggs for stem cell research? And if so, what are the requirements that person must meet in order to do so?
Meaning X, women X.
Yeah.
I mean, you could find some stem cells in there, but remember, eggs have only half the genetic material for a human being. So to answer to your question, no, not necessarily.
Uh, all right, thank you. So right now, I would like to invite Bianca Jimenez, she's the chair of the MBS chapter, student chapter of Tech Costa Rica, to continue with with a couple questions.
Mhm.
Thank you, Ian. Can you hear me? Can you hear me clearly?
Yes.
Okay. Thank you, Ian, for the introduction, and thank you, Dr. Masan, for being here and all of our guests. So I'm going to continue with the questions we have from the Costa Rica Institute of Technology. These are kind of basic questions. The first one is, are all stem cells the same?
No, all stem cells are not the same. There's some. James, can you answer to that question? Because I mean, well,
So,
let me answer it. Let me answer it. No. All the stem cells are not the same. Stem cells can be multipotent, pluripotent, induced pluripotent. I mean, they're different cells depending on what their capabilities are and the actual CD markers they got. So, say the mesenchymal stem cells itself are multipotent. That means that they can only convert to certain tissues. Although pluripotent cells can convert to many other tissues. And if you differentiate those into, you can actually differentiate pluripotent into specific tissues, you know. So to answer to your question, no, all stem cells are not the same.
Okay, thank you for the answer. Next question would be, are the cells differentiated into specialized cells before being injected to the body in case they would be injected to the body?
Well, yes, it depends on the disease again. I mean, if you have osteoarthritis, for instance, which is bone cartilage degeneration in your knees or in your hips, you want to inject already differentiated cells, chondrocytes, which is the cartilage cells that are, the compound is the biggest component of cartilage. And some other diseases, you might want to do different injections, localized injections. For instance, for spinal cord injuries, we've noticed great improvements if you do neurocytes, already differentiated neurocytes. And for cardiac disease, say a patient had a heart attack, we're already doing catheterism procedures where we inject cardiocytes inside of the muscle, the heart muscle, where the heart muscle, regenerates. Which the two tissues that historically we haven't been able to regenerate is our brain, spinal cord, and Becca, you want to go ahead with your question?
Yeah, actually, I had another question about this same topic, and this is, how do you induce the stem cells?
James, would you be so kind?
Sure. It's through using other growth factors, very similar to the way that these vaccines are. We use other growth factors from the cells that we want it to be to get to the nucleus of the cell that we're trying to differentiate.
Oh, I understand. Thank you for your answer.
Sure.
Next question would be, can cancer be treated or studied with stem cells?
Well, cancer is a completely different animal, to say the least. But through genetic, go ahead, James.
So cancer is a completely different animal. There are very specific types of that, such as natural killer cells, NK cells, and dendritic cells that are wonderful in treating that. But it's a very specialized field.
So basically, to finish your answer, Bianca, we can differentiate the MNCs into natural and NK cells, natural killer cells, and there's some other people around the world doing gene therapy, which is basically using natural killer cells. So
I understand.
Yeah.
So okay. So the next question would be, how are the cells delivered to the right part of the body?
It depends on the disease. if you have Alzheimer's, we would like to do it intrathecal, right inside of the spinal fluid. Spinal fluid surrounds the brain and the spinal cord. So neurodegenerative diseases would like to put the cells inside of the spinal fluid. Some other scientists use mannitol, which is a diuretic, in order to open the blood-brain barrier. And stem cells are large, right? And in order for the stem cells to go into the spinal fluid, we're currently doing intrathecal injections, so directly to the spinal fluid.
I see.
And for instance, you chondrocytes or even mesenchymal stem cells that will differentiate into chondrocytes for osteoarthritis in patients' knees or joints. You want to do intra-articular fluoroscopic guided or ultrasonic guided injection.
I see. Thank you. Okay, next question is, if the cells are not my own, how will my immune system be prevented from reacting to the transplanted cells?
The stem cells are very unique. They're immunologically privileged. It's not like a liver transplant or even blood transfusion from a different kind of blood that you have. So they don't have HLA, which is the antigens and surface antigens.
Mm-hmm.
There's no odds of rejection by doing a stem cell transplant from, say, umbilical cords or some other tissues.
Thank you. I think we had a couple of questions in the chat. First says from Maria del Pilar. Hi, I'd like to know if you've had cases of recovery of bone tissue in the jaws with stem cells.
Yes, absolutely. Not only in the jaws, in the jaw. I mean, in every cell. Bone is one of the tissues that we've been able to differentiate from mesenchymal stem cells, and yes, we have. We had a patient called Anthony Chicales. It's in our website and all that, who had a gunshot in his left leg. We did a transplantation of stem cells, bone cells, basically. And after 6 months, his orthopedic surgeon said, there was no. Yeah. Okay. So to finish to answer to your question, we could do jaws. We're already working with dentists in order to build up bone before doing implants. Whereas people need to wait 6 to 8 months before the implant, they go bone in less than 4 weeks.
Thank you. We have another question. This is an unidentified username, but it says, can stem cell be a treatment for fibromyalgia?
Yes, it can. It depends on the patient's clinical scenario. Since we don't have the name of that person, I'm not going to go any further with that.
Next question says, can SCT increase chances of cancers cause development of teratomas?
No. The answer to that question is no. Teratomas are benignant tumors. Terocarcinoma is a malignant tumors. Teratomas can have bone, hair. It's a kind of weird tumor. But the odds of that to happen, some studies have said that pluripotent cells, because they can convert to any tissue, might have a higher risk to develop teratomas, but we haven't seen one in our practice for years. I mean, I personally have never seen a teratoma development or any other kind of cancer, quite frankly, in any patient.
Yeah.
All right. So, excuse me. So before continuing with the questions in the chat, I would like to use the PowerPoint presentation that Dr. James and Dr. Paven has prepared for us. So I'll proceed in sharing the screen, and then we can continue with the flow of the chat.
Thank you.
James, go for it.
Okay, wonderful.
Sure.
We're just waiting for the screen to load here.
I'm
So,
gonna
Okay.
stop my video real quick, but I'm gonna be on in here, right?
It's all right. Don't worry.
Beautiful. Go ahead.
So,
So
so
you
here
guys
we
did.
here we are at the lab for the Stem Cell Transplant Institute. This is where we make the cells. This is where they originally started in umbilical cords, and then we differentiate them to all different types of amazing, fun cells right now, such as cardiomyocytes for the heart, neurocytes for the brain, and of course, mesenchymal cells, which are most commonly used. Some of the, as you can see behind us, this is some of the equipment that we're using. We got quite a big lab here. And then actually our PowerPoint, Professor Pavan here is going to take over and go walk us through the PowerPoint.
no,
Just going to be
absolutely.
some initial steps on what are stem cells and how are we using them now.
Sure, absolutely. Thanks, Ian. If you could go to the next slide. I have some basics too. If anyone can stop us anytime if you have any questions. You can go to the next one. I had a quick outline as to what we're going to talk, but real quickly, yeah, just the one before. What are stem cells? We all know that they can divide and renew long periods of time, so they can multiply and proliferate multiple times. And some of the stem cells, especially embryonic and pluripotent stem cells, they can divide for more than 100 to 150 passages or even more. But multipotent stem cells, probably at least maybe like 8 to 10 passages before they slowly start senescing or trying to reduce their proliferation rate. And the stem cells are very unspecialized, meaning they can give rise to almost all tissues of the body. And right from heart, liver, as Dr. Meeson said, all muscles and kidneys and heart and the brain cells, neurocytes, cardiomyocytes. So it's very, very potent and powerful, which is really nice. And these are the cells which actually make the baby. The entire human body is made from a group of stem cells. And that is the cool thing. And now, since we know how these unspecialized cells give rise to specific cells in the body, we can now use specific growth factors, as Dr. Phillips said, to turn every cell to whatever we need, like brain cells or cardiac cells, so we can actually train these cells to become that. So you want to go to the next slide, Ian? Yeah, so Dr. Leslie Meeson already told that different types of stem cells. So a quick diagrammatic representation. So totipotent is the zygote, which is nothing but the first cell which is formed when the egg and the sperm fuse. So they can, of course, give rise to the entire human body, which is cool. But we don't use those for any treatments. And pluripotent, on the other hand, it's embryonic stem cells and also induced pluripotent stem cells. So we have been using this. Of course, we directly do not inject pluripotent stem cells into humans. We usually differentiate them and then we can use them for therapies. But the most coolest cell which we can use now is the multipotent stem cells, which are the adult stem cells. So the mesenchymal stem cells come in this category. And many of the questions we saw was they had concerns with teratomas or any cancer formation. So multipotent, as of today, I don't think we have had any issues with forming any cancers or having any crazy side effects. So these are the best cells as of today we can use. And then, of course, unipotent are the skin cells and the differentiated cells like blood cells and the epithelial cells. So these are just the different types of stem cells which exist right from birth to the end. But multipotent stem cells, or the mesenchymal stem cells, are the best ones. So yeah, next one, Ian. And if anyone else have any other questions, please stop us and ask us if you have any other questions. So I went through a few of these cells in detail. So you can go to the next slide and see if there are any. There it is. So just a nice diagram. So that's the blastocyst, which is, say, it's like a 7 or 8 day old human embryo. And you can see that the red circle is where your embryonic stem cells are. So those are the cells in the red circle which actually give rise to the entire human body. This is really cool. And I think in the 1990s, early 1990s or 1995-ish is when we successfully isolated these stem cells. So these are pluripotent stem cells, embryonic stem cells. So the next one is, oh yeah, just quickly to show you. So it's around day 6-ish, the blastocyst. So that's where we get most of the embryonic stem cells from. And most of these cells, especially for research, we get them from donated in vitro fertilized embryos, which is good. So it's not from aborted fetuses or anything like that. So it's mainly in vitro fertilized ones. So let's try the next one. Let's go to the next slide. And these are mesenchymal stem cells which we are using right now. So you can see at the lower left, that's the umbilical cord, which is like a white tube which connects the mother and the baby. So we cut them, and of course it goes through a whole ton of regulations, right from bacterial, fungal. So it has to be negative for all the viruses and bacteria and fungus. And we also see the genetics of the parent up to 2 or 3 generations to make sure that they do not have any genetic diseases. So it has to be absolutely clear of any diseases and any pathogens. And these are the only cells. Then we start to culture them. And then it usually takes like a couple of weeks or more, sometimes 3 weeks, for the cells to come out of the cord and slowly start to grow. So we try not to forcefully push them out. So they come out naturally by themselves. And these cells have been very, very potent and very nice. So that's the cord. And those are the umbilical cord stem cells, tissue stem cells, which you see on the right, lower right. And that's how they grow in a dish. And we keep track of these cells all the time, 24/7, to make sure they're growing well and they're healthy and there are no issues with the growth and proliferation, division rates of these cells. So let's try the next one. And we also have some cells, as Dr. Mason also said. There are cells in every organ in the body, like hematopoietic stem cells, like blood stem cells, bone marrow stem cells. We also have cardiac stem cells, and we also have brain stem cells, kidney stem cells. The only thing is, with time, these cells do not respond to an injury. Even though they're there, they're just sitting around. And even if there's an injury for the brain, the neural stem cells, sometimes they do respond, but as you age, they stop responding. And this is when you need a boost of fresh stem cell treatment and stem cell therapies. So with time, hopefully we'll get even more better in differentiating these cells, especially the mesenchymal stem cells. So let's try the next one.
Wait, I have a question on this one.
Yes, yes.
Why is it that the body, after aging, the stem cells just not decide to do their function as in their early stages?
Absolutely. So when you are young, there is less damage to the DNA. And as we age, there is higher DNA damage seen in the DNA, higher damage seen in the DNA. So approximately every time the cell divides, there is at least 1 or 2 errors which naturally occur. It's a natural evolutionary trait. So say if the cell divides 50 times, so that's almost 100 to 200 different types of permanent damage to the DNA you're seeing. So more damage, the slower and unhealthy the cell becomes. And so that's one big reason we age too. And humans also, we also have many of the species, we do have DNA repair mechanism. So they also repair the damage, but they can only do so much. So if there's too much damage, and of course, environmental factors play a big role. Your diet, if you smoke or consume alcohol, or environmental factors, all of them play a big role to see how much damage is caused in the cells. And the cells can repair most of it, but sometimes even the repair mechanism gives up and it says, oh, it's too much, I just can't deal with it. So yeah, that's a nice question. So usually the repair mechanisms around age 40 to 45-ish or 50-ish, they tend to slow down. So this is the time, I guess, more diseases start to show up in humans. So there's a nice correlation we have seen with that.
And then I have another follow-up question to that one.
Sure,
Since, well, in stem cells therapy, I'm introducing stem cells that are, they're barely getting used to my DNA. So isn't that like a problem for the immune system? Like, do they get like detected easily because they're not as aged as the new stem cells? Is there gonna be like a problem in my body, or how does the immune system treat these new stem cells?
Correct. So that's why that's the question on immune evasion. The immune cells should not detect the new cells. So umbilical cord stem cells are the best cells to use, and it can be used for everyone because these cells do not, as Dr. Meeson told, the cells do not have protein, the surface receptors on these umbilical stem cells. So the immune cells cannot recognize them. So like, just say, like if I have to recognize you, Ian, you have different features, like you have hair and you have the nose. So I can recognize you the next time. But umbilical stem cells, it's blank. It's like a blank face. So the immune cells cannot detect them. So they think umbilical cells as a body's own cells, so they don't attack them.
All right, thank you very much.
Yeah, no problem. So the surface proteins are, especially it's called the human leukocyte and MHC antigens. So they are extremely low expressed or almost absent.
All right, thank you.
No problem, no problem.
Should I keep on going with the next slide?
Sure. Yeah, I have some more slides. Anything to add in for the mesenchymal stem cells?
Not really. I'm looking at Dr. Yamanaka from the IPS, so you can give. I know him in person. He's a great guy.
He's a super great. I know. Yeah. So he's the one who won the Nobel Prize in 2012 to actually make induced pluripotent stem cells. So this is where you take like a skin cell or a blood cell. So you cannot use those for therapy. So you can take a skin cell from one patient, and then you can reverse it or reprogram it back to a stem cell. And once they become a stem cell, now the stem cell can give rise to all the tissues of the body. So this is a great treatment for personalized medicine. It cannot be used, of course, for all other patients. It's only patient specific. So that's one drawback of iPSCs. And of course, it takes a little long time. It takes almost like 2 months to 1.5 to 2 months to get the iPSCs, and then maybe another month to 1.5 months to make the cells which you want. So it's almost like a 3 or 4 month procedure, but it's doable. This was the first iPSCs were the first ones to actually show that all the cells in the body have the capability to be reprogrammed back to a stem cell. So which is really cool.
So if we can reprogram our old cells to become a new stem cell, is this is this reprogrammable stem cell as effective as a new stem cell?
It is very 95 to 98% is my guess. I can never say 100%. So there's still small differences for sure.
Yeah, well, 95% is pretty good.
It's pretty good. Yeah, just like the vaccine.
Yeah.
So, but yes, they do show properties of new stem cells. Or they behave very similar to the stem cells in an embryo, and they differentiate very well too into different tissues of the body. But the only thing is, you can only use it for that person. So making it the cost might be a little more expensive, and it's a little more laborious to make those cells, and it's time consuming. It'll take like 3-4 months to get those cells, and it's not all the time you can get cells from every patient. It just depends on the genetics of the patient too. If the genetics of the patient is pretty bad, if there are too many diseases running, then that's going to be trouble using cells from the same patient. So it's good to use mesenchymal stem cells rather than iPSCs.
All right. So if it's only used for people, for that same persons, what happens if we have somebody with very similar DNA? I'm just gonna say, throw it out there
Yeah.
in the dark, like, I don't know, a twin.
Twins, it is possible because they share pretty much the same. If it's identical twins, yes, they will share. The genome is exactly the same. But if it's fraternal twins, there might be slight differences. It's called epigenetic differences. So there are very, very slight chances of rejection, but very rare. But yeah, twins is fine. But from even the same person in the family, sometimes it's difficult to use iPSCs. So that's why mesenchymal stem cells is the way to go.
All right, thank you.
No problem. And let's see if I have a few more slides after that. I also have, let's see, iPSCs. Can you go to the next slide, Ian? Oh, yeah. So someone asked a question about stem cells from the egg. So Dr. Meeson already said, yes, it might be possible, but I don't think we can use those cells. It's very, the great point Dr. Meeson told was there's only half the genome in the egg. So it's very difficult to use that because you need one full genome from the mother and the father. So you need the cells which make up the entire complete genome. So it's very difficult to use cells from the egg. But there are various other cells which we also use, bone marrow adipose, which you said, and then the dental cells, then iPSCs, endometrial adipocytes, of course, and then all these have the capability to differentiate into different parts of the body. All are mesenchymal stem cells, but the best again to use, and the best cells you can use where you can grow in large quantities is the umbilical cells. Because if you see a bone marrow, all the other cells, mesenchymal stem cells, is the age of the patient. So if the patient is 50 years old, the bone marrow cells are 50 years old. Even the adipose is 50 years old. So the cells are already old, but umbilical cells is day one. It's brand new and fresh. So you cannot get any more newer cells than the umbilical cord tissue cells.
Yeah, so just taking a wild guess here. So what happens if we already discussed that having just egg donors is not good enough because we only have half of the information. What happens if we have by separate means, egg donors and sperm donors, so we can have the full zygote? Is that I don't know, biological, ethical to do?
Well, you have so that's in vitro fertilization, where you can actually there'll be a sperm donor, and then you can get the egg and you fuse the egg and the sperm in a dish under the microscope, and once it grows for probably 3 or 4 days, you transplant back the egg and the sperm, which is fused back into a surrogate mother, into a mother who wants to carry the baby, and then the baby grows. But those cells are completely different. There you're actually growing a new baby rather than.
Can I interrupt? Yeah. Can I interrupt right there, Ian? Ian, so that right there, you're talking about in vitro fertilization. getting a sperm and an egg. Yes, you got the 46 chromosomes, but yet you're creating a new human being. You know, once the zygote is ready, it's a human. And so there's ethical, there's religion, there's a lot of things involved. And at the end of the journey, you're not looking to get those cells because they're super undifferentiated. They're pluripotent indeed, and there might that's where those teratomes that kind of cancer that the other lady was making a point of might happen. So why would you do that instead of just grabbing the umbilical cord itself that's full of stem cells and grow them from there? I mean, at the end of the journey, we're looking for the mesenchymal stem cells, and we can differentiate them from there. You know.
Can I ask a question? I know we've been talking about aging of the cells as we are expanding them, they multiply. And how many multiplications? Like I know the telomeres are going to get damaged and the DNA gets damaged at each, you lose DNA. And so how many divisions and how many expansions, at what point do you say right now it's safe? And, you know, at what point do you say this stem cell might not be safe to use? You know, it has undergone so many divisions.
That's a great question.
Yeah.
You're completely correct about that. Through every differentiation, you're going to lose a little bit. So in our case, because we strive to create the absolute best cell in the world, we use passage 1, P1 and P2s.
That's it.
They start at P0 directly from the umbilical cord and we go to P1 and P2. Now, a cell, to answer your question as well, can differentiate up to 20 times. Yeah. But we stick
some clinics around the world, like in Thailand and India and some other places, they do P20. You know, they differentiate themselves and culture expand them, 20 times just to get the most of it. But they lose, not only they lose their qualities, but they don't after phase 7 of the culture and they just don't replicate as good. So this said, we don't do more than phase 2.
So question would be that, do you guys, is there a process to monitor if any mutations have occurred during the process of expansion? Do you routinely do that, or is it not routine?
More than routinely. Yeah. It's all we do.
Every day.
Excellent. Thank you.
Endotoxin microbia, genetic testing. I mean, there's a lot of things that, I don't think it's this conversation would last for 20 hours if we start doing that. But yes, of course.
Yeah. You could show slide 18. Can you go to slide 18?
I cannot see the numbers, but you tell me when to stop.
Okay, keep going. Jeez. Oh, sorry. It's
Did I miss it?
Hold on. It's after. Oops. After generating iPSCs, it's the 4th slide, I guess.
This one.
One more back. Sorry, I went off to this one. There you go.
There we go.
Yes.
Yeah. So these are all the 46 chromosomes in the cell. So this is a representation of every chromosome. So you can see chromosome 1 is 2. Chromosome number 2 is 2, chromosome number 3 is 2. So all are pairs. So each chromosome comes, one from mom, one from dad. And we can actually check for mutations and deletions, duplications, and all of that just by looking at the set of chromosomes. So we make sure the cells are absolutely 100% healthy.
All right, thank you very much.
No problem.
So should I keep on going with the slides?
Yeah.
Yes, go ahead.
I don't know, did someone else have any other question?
Yeah, and I think the University of Costa Rica have some more questions. They had like 25 questions, I believe.
Oh, yeah, sure. Yeah.
Yeah, we have a couple questions from the UCR, as Dr. Masen just said. If you just allow me to stop sharing, please.
Sure.
Okay, so I need access to those. So the questions for the UCR pharmacies department, they're gonna be used maybe to enable to potentiate their research paper that they want to publish. So they had a couple questions regarding the Parkinson's disease treatment specialized with stem cells. Like if you guys do this kind of research and if you could elaborate with it.
Yes, we do. We're currently conducting a study with along with the UCLA for Parkinson's disease. You know, it's one of the neurodegenerative diseases that we're treating, and we're having great results with both neurocytes and mesenchymal, or I should say, pluripotent cells injected directly into the spinal fluid. There's something that's called cell signaling. Any damaged cell in your body sends signals, inflammation signals, to say the least. And those are the only the markers that stem cells read, and they will go to the damaged tissue and do a physiological process called homing effect. So they do home in the areas that are affected, for instance, in Parkinson's disease, and start the regeneration process. We've seen Parkinson's patients, taking amazing doses of levodopa, which is the only medication that stops tremor, and as well as Parkinson's, Alzheimer's, ALS, multiple sclerosis, neurodegenerative diseases. We've seen great results with this kind of the same protocol.
All right, and is this the Parkinson's disease right now like more like in the kind of a ongoing research phase, or is there like right now like something that works and we're just sticking with it right now?
There is, as I said, we're on an ongoing research with along with the scientific department of stem cells with the UCLA. Dr. James is involved as well, and some other people. So there is ongoing research. Not only us, I'm sure there around the world. There's plenty of investigation being done.
All right, so we already spoke about the pathologies that they treat. So I think this question goes a little bit more focused to the Stem Cells Transplant Institute, and they would want to know if Costa Rica if the studies are being done here in Costa Rica, and if we have Costa Rican test subjects.
We do. We're at this point we're working with the bioethical committee to do some patients. We're doing compassive patients, but the clinical trials we're about to start within the next week or so. It takes a lot of paperwork, a lot of money for many different things, as you guys know. And yes, we're doing clinical research here. Yes,
With people from Costa Rica.
correct. Costa Ricans. Yes.
All right, and have the Costa Rican social. Excuse me.
We have a group of neurologists, pathologists, neuroanesthesiologists. I mean, there's a big group going on. But unfortunately, to do a clinical trial, well, first of all, you cannot charge a patient anything, and you can definitely publish the results, which is something that we're doing. And yes, there are Costa Ricans being treated at the moment. Yes.
All right, thank you. And have you guys received any interest or approach from the Costa Rican Social Security Fund or Caja del Seguro Social to invest in the future of this type of treatments, or is it just more like a private?
It is wholly private. Believe it or not, the Caja, the CCSS have a big bank at Mexico Hospital, Mexico, for stem cells, but they don't know how to use them.
Okay,
So there's a big bank. They're storing stem cells every single day, but they don't really know how to use them, which is sad. But I mean, I even spoke to the Minister of Health to be in this meeting, and the president and some other authorities, but they just want to avoid the subject. and as well as in Costa Rica, Ministry of Health, FDA in the United States, some other authorities, regulating authorities, they just don't know about it. You know, as science continues to go on, they just don't know what's going on. So there's people in the hospital in front of a computer being asked if they should be give permissions for this or for that and what it is, but they don't know about it. And as we're doing cutting edge science, we personally try to teach them how it works, so they're more suitable to pursue a better an easier way for physicians and clinicians and scientists to approach treatments on this matter. In fact, the FDA have just banned culture expanded mesenchymal stem cells in the U.S. and even exosomes that are healing substances that stem cells in the culture media release, which is kind of a downer because they don't know any better, you know. And that's my perspective, my personal opinion. There's a lot about big pharma involved. You know, once they see that something is working for X disease, they will try to stop. They will try to stop it, and that's the bottom line. You know, but that's my personal perspective. Is an unbiased perspective, and if you share it, you share it. If you don't, you don't, and I respect everyone.
All right, thank you very much. The next couple of questions, I think they come a little bit they come like in a pair, because then you just mentioned that the CCSS has a big bank in Mexico, but we as a country are having
Mexico Hospital.
Mexico Hospital, sorry. Okay, but we have a big problem in actually getting to use these resources that we have. Do you think the challenge that we are presenting is a little bit more like technical on the medical field, or is it more like according to the laws?
It's definitely the law. Ministry of Health, they don't know how to do the law because they don't know anything about stem cells. That's period. I mean, same as FDA, same as as the science as us scientists continue to research and find out more things to do, they are just stepping behind. I mean, they're just left behind because they don't know. They don't have. There's no such a thing as a scientific stem cell department of Costa Rica making research with the government funded government funds. So that's what's going on right now as our legislation, our country, and it's even worse in other countries, such as the United States and some others, England, London, whatever. But that said, it's frustrating sometimes, looking at the Minister of Health coming into your office and asking for things that they don't even know what they're looking at.
Yeah. So thank you very much for your answer. So the second question would be, right now, do you think your patients at the clinic, are they mainly Costa Rican, or do we have a lot of people coming for medical tourism?
99% of patients come from the United States, Canada, and Europe.
Okay, so we can see that there's actually not only a benefit from a scientific and research point of view here in Costa Rica, but also there's a market out there, so we can become one of the leading countries, at least in Central America, to implement this kind of treatments.
Absolutely. We're already top 5 medical tourism destinations in the world. So yes, to answer to your question, we can definitely be one of them. Yes.
All right. So we have a question also from part of the faculty of UCR, of the pharmacy faculty of UCR. What is the estimated price of the treatments, stem cells treatments? I know it varies a lot from the kind of disease and condition we are treating, but just to throw a number.
Pricing, it depends. It depends on how many cells are you going to use, how specialized the cells will be. So I cannot really give you a ballpark of cost, which again, we're not talking about prices or money here.
Really. Yeah, I get it. Don't worry. So I think we have a couple more questions from we have the MBS. I don't know if Bianca wants to take them, or should we take the chats? you guys decide?
Chats are a little. There's a. I see a bunch of pictures. I already telling. I'm asking everyone to send the answer, the questions to our main email. So because otherwise this is gonna take forever.
Yes, it would. I agree. I agree.
Yeah. So those questions will be asked. Will be answered for sure. Dr. James, do you wanna, or Dr. Reginelli, do you wanna add anything else to the conversation?
We did. We just had a couple other things. Did you want to go over any of the slides or anything, or were you done?
I think that's
okay.
It, yeah.
So again, not all cells are different. In administration of each type of cell for each type of condition has to go in a different place. Nothing is the same with what we do. It is very specialized, and so everything now has a place for it. So you can't just throw mud against the wall and say, okay, we're going to try a stem cell treatment. It's really getting to a point of being very specialized now. So that's where, at the Stem Cell Transplant Institute, where we really excel at that specialized medicine, and really taking it a step further in understanding the processes of what we're doing in these type of cells. We are making and working with the best cells in the world, different types. So it's always ongoing and always learning. So we don't get left in the dark either when somebody else creates or invents something new.
Correct. And just to add a point, umbilical cord tissue stem cells are the way to go. So there are a few clinics which might offer bone marrow and adipose, but you don't want to go through the pains of surgery. And all those are invasive and it's a mess. So this is and less potent. Those cells are less potent. So umbilical cord tissue is non-invasive to isolate those cells. And they are the most potent. So that's the best cool cells to use for treating most of the diseases here.
So with that said, we're going to get back to work on these cells here at the lab. We really appreciate you guys letting us come out of the dungeon for a few minutes and be on TV. So thank you. But we got some more work to do, and it was wonderful. Please, any questions, send them along, and we are happy to answer them.
Perfect.
So,
yeah, thank you very much for joining us. I really appreciate you guys coming in, even in the lab and wearing the scrubs. That means a lot to us.
We're working right now, so we stopped what we're doing to turn around and do this. And
so you guys see in the back of the doctors, there's of cells, live cells right there. You see that? That's the microscope. Yeah. So yeah, I'm sure you're working. Same as I am, there's a couple of patients that I gotta go treat. But if might there be any other question, Ian? Doubts?
I think we cover almost about it all. I know we're running out of time. Also, I want to thank you guys again once on behalf of the Secrets and Systems Society and the Engineering of Medicine and Biotechnology of IEEE of Costa Rica Institute of Technology. It's been a pleasure working with you guys, and if it may be in the future we can have another questions and answers straight with the public.
Absolutely.
Yeah.
Just manage for more scientists to be around,
Sure.
because we can discuss a lot of things, and we'll go from there.
Of course. So thank you very much. I will not end the meeting yet because I don't know if they want to get the emails, but you can disconnect already. I'm just gonna project the slide we had at the beginning. That's gonna with the QRs of both the clinics.
Mm-hmm.
So
sure.
I think we're good to go.
Yeah, and if you want, I can stay for you I can bear with you for a few minutes, for a few more minutes in order to answer anything else that you might have. But James, Dr. Evan, I'll see you soon. I'll see you on Friday, right?
That's right.
Tomorrow? Jesus Christ, time goes so fast. I'll see you tomorrow, man. Take care.
Yeah,
Bye-bye.
take care.
Bye-bye.
Thank you very much. Bye.
Bye-bye. All right, Ian, so anything else you might want to add? Questions, answers?
Let me just check. Have a quick check right here to see we cover almost.
I have a last question from MBS. Just to finish with the topic. Besides the issue of law, can you give a point of view towards an improvement in stem cells, worldwide research and applications in medical treatments?
Excuse me. Can you repeat that question?
Yeah, of course. Besides the issue of law you have right now with the use of stem cells, can you give a point of view towards an improvement in the research of stem cells worldwide and applications in medical treatments?
The only possible scenario is where governments start funding clinical trials, which is I can see that that's not going to happen anytime soon because of the big pink elephant behind the scenes called big pharma. You know, during Bush administration, the FDA banned every single funding for stem cell research because they started finding that they were working basically. So to answer to your question, the only possible way that I can see this happening would be governments opening up research, funding for universities and institutes like us for research.
I understand. Thank you, Doctor, for
Well,
your answer. And from MBS, I want to say thank you for attending this panel discussion. It was very refreshing, and so that's what we have to say.
you're very welcome, Bianca. Nice meeting you. Anything else, Ian? I'll let you finish.
Yes, we have one question from Kelly Lockwood. I invite Kelly to please open that microphone.
Okay.
Thank you so much. And I apologize, I was only able to catch the last 15 minutes because I too was teaching. But Leslie, we have been to Costa Rica
Hi,
for
Kelly. How are
a
you?
stem cell. Very nice to see you. And
Yeah.
I have a quick question because I just heard a little bit of one of the other comments, and that's 5 years ago when you did a stem cell transplant on my brother, we did use adipose tissue. Has there been further work or research to say there's a better source of cells? Is that what I heard in that last conversation? I just wasn't sure.
That is correct. That is correct. Back then, we were in the very initial stages of culture expanding stem cells from umbilical cords, and that's why we were obtaining the cells from fat and bone marrow. But that's old technology at the moment. Although your brother had good results, as far as I understand, as far as we agree on that. There are much better cells right at the moment. We are actually differentiated mesenchymal stem cells delivered from the culture expanded part of the umbilical cords to different cells, heart cells, brain cells, bone, skin. I mean, different cells. So to answer to your question, yes, there's much better technology nowadays that we work on and research on. Yes.
Thank you for that answer. And you're very correct. We had a very, very positive experience. And we look forward to connecting with you again post-COVID.
All right. Thank you very much. I look forward to seeing you guys in Costa Rica once again.
Thank you.
All right, Kelly. Ian?
I don't have any more hands that are raised right now, so I think we're good to go. Thank you very much. Once again, Dr. Mesen, it's been a pleasure to have you here and answering the questions.
You're more than welcome, Ian, and from all the team, the University of Costa Rica, Institute Tecnological Tech Costa Rica, and other universities that have been connected through us. I appreciate your attendance, and we'll look forward to seeing you soon with more information.
All right. Thank you. Have a good day.
Have a good one. Bye-bye.
You too. Bye.